What happens inside a cryochamber during cryonics? Imagine a future where people might be brought back to life after death. Inside a cryochamber used for cryonics, this idea becomes a carefully planned process. When someone is declared legally dead, the first step is to keep their brain and body as stable as possible. Medical teams supply oxygen and blood to the brain to keep minimal activity going until they arrive at the cryonics facility. Once there, the body is cooled slowly by wrapping it in ice and lowering the temperature gradually. This slow cooling prevents shock and damage to tissues. The main part of the process involves replacing the water inside the body's cells with a special chemical called a cryoprotectant. Think of it as an antifreeze for the body. This solution is usually glycerol-based and is carefully injected into the blood vessels. Its job is to prevent ice crystals from forming inside cells, which could tear apart delicate structures. Instead of freezing into ice, the tissues undergo a process called vitrification. This turns the body into a glass-like state, stopping all biological activity without damaging the cells. The temperature inside the cryo-chamber is then lowered to around -196 degrees Celsius or -320 degrees Fahrenheit, which is the temperature of liquid nitrogen. At this point, the body is in a state of suspended animation. Biological decay is halted, and the body can stay preserved for many years. Unlike quick cold treatments used in sports medicine, cryonics involves a slow, controlled cooling process that keeps the body intact. This process requires precise control because any mistake could cause damage. The goal is to keep the body's tissues as close to their original state as possible. The cryoprotectant is vital because if water froze inside cells, it would expand and cause cell rupture. By replacing water with the antifreeze solution and vitrifying tissues, the body remains in a stable, glass-like state. In essence, inside a cryochamber during cryonics, the body is carefully prepared with cryoprotectants, then cooled down to cryogenic temperatures. This halts all biological functions, preserving the body in a way that might allow future revival when advanced technology becomes available. It's a fascinating mix of science and hope, combining chemistry, biology, and engineering to keep the possibility of life after death alive.